Good morning. Today I would like to talk about this paper titled Gel-Free Single-Cell Culture Arrays on a Microfluidic Chip for Highly Efficient Expansion and Recovery of Colon Cancer Stem Cells. As you know, it is quite difficult to form spheres from tumor stem cells, but with this approach, they really improved the expansion and recovery of these tumor spheres derived from single cell. And how they do that? They built a microfluidic device. As you can see here, it contains 3 layers, and in the second layer is where the main channel is located. And here you see that there are different branches, and they are going to be able to capture 16,000 cells. So first, they prepared an alginate solution, an alginate suspension with these tumor stem cells. And the cells are going to be captured in these microchambers. So first, they had to find the best concentration. So they modulate the cell density so that just one cell stays in this microchamber. Once they found it, then they have to enclose these chambers. So they use a chitosan infusion so that the cells are enclosed here. And because chitosan layer can be destabilized, it's also impossible to recover these tumor spheres. And they show here how the cells are trapped in these chambers. As you can see here, the bright film, and the cells are marked with the cell tracker, the red cell tracker. And you can see here how just one cell is trapped in these microchambers. They also show that the cells that they enclosed have different sizes. And here's how they modulate the cell density so that they have to balance between the cell densities that produce no cell, single cell, double cells, or multiple cells. So they choose one 10 to the 5 cells per milliliter, and that's the optimal concentration in order to have them in most of the single cells. Okay, so they measure the cell viability before the input, after the input, and also after recovery of cells. They also measure the cell diameter in these 3 stages. And here we can see how they filled the microchambers with the alginate solution. And the excess of the alginate solution in the main channel was expelled by the airflow. Here we see that the microchambers were enclosed with this chitosan layer, which was mixed with the FITC fluorescence in order to be able to see it. And then how this layer can be destabilized by air infusion. Okay. So here we see the cell viability in the time course, how it decreases. That's why the experiment lasts 7 days. They also compare this gel-free approach when they use hyaluronic acid. So here you see with hyaluronic and without it, and also gel with hyaluronic. And there is a statistically significant difference when you use these gel-free hyaluronic. So this is the best way to produce the tumor's first cells, because when you use a gel, it limits the expansion of the tumor cell. That's why when it's a gel-free approach, you eliminate this problem, and then the tumor sphere can expand. And the hyalurona helps to obtain these tumor spheres. So here we see the consecutive imaging. This is how it began at the day 0, and then how the tumor sphere is formed until day 7. In the first row, you see the single cell derivative tumor spheres, and in the second row, you see the multiple cell derivative tumor spheres. So here they show before the recovery, how the tumor spheres are located inside the microchambers, and after the recovery, when they use the reverse flow, the microchambers are empty. And they also use live dead kit in order to check the viability. And as you can see here, the spheres, the cells that form the spheres are alive. They also perform the cytometry, and they measure the expression of these biomarkers, which are specific for stem cancer cells. And you see that the expression, the proportion of these markers is positive. And finally, they perform experiments after the recovery of the tumor cells. They wanted to know if they are able to differentiate into multilineage. So they confirm that the billing expression suggests that the B cells have differentiated into enterocytes, and also the CK20 shows that they are going to differentiate into epithelium, and so the stemness is confirmed from the recovered tumor spheres. And so they propose this microfluidic chip, which is really useful because it eliminates the factors that decrease the viability of the cells in comparison with the microdroplets approach. And so this is a great improvement for this science field. Thank you very much.